Fine. I'll begin. Okay. General and cellular physiology, we have got cell organelles and their function. Plasma membrane physiology is a very important aspect you need to learn, especially the transport across cell membrane. Cellular receptors, there are four types of cellular receptors: type 1, type 2, type 3, type 4. Type 1 are called as inotropic receptor. That means they are ligand gated ion channels. Then metabotropic receptors, otherwise called as G protein coupled receptors. Type 3 are tyrosine kinase mediated receptors, and type 4 are steroidal receptors or intracellular receptors. This you might have learnt in biochemistry, especially with cellular mechanism of hormone action. You will be learning it in physiology also. Okay. Then comes membrane potentials. There is something called as Nernst equilibrium potential. Sometimes MCQs are asked. Nernst equilibrium potential for sodium is, it is plus 61 millivolts. What is this concept? Whenever you allow movement of single ion through membrane, how much will be the membrane potential at the equilibrium concentration in ECF and ICF? So if there is equal concentration of sodium in ECF and ICF, what will be the membrane potential? That is Nernst equilibrium potential. So Nernst equilibrium potential for sodium is plus 61. For potassium, it is minus 90 millivolts, right? Then we have concept of resting membrane potential. So resting membrane potential is a characteristic feature of every excitable tissue. Every excitable tissue has got its own peculiar, very peculiar resting membrane potential. For nerve, it is minus 70 millivolts. For Purkinje fiber and ventricular cardiac muscle, it is minus 90 millivolts. For SA node, it is approximately between 50 to 60 millivolts, usually considered as minus 55. For smooth muscles, it is also higher. Please remember, it is higher for SA node and smooth muscles. For smooth muscles, it is usually considered as minus 50 to minus 60. If was one single value you have to give, then usually consider minus 56 millivolts as the standard value for smooth muscles, resting membrane or membrane potential of smooth muscles. Then body fluid compartments, as we all know, there are two compartments: intracellular, extracellular, and in between these water is distributed. We all know there is 60% of water in our body, and we have got approximately 3 to 3.5 liters of plasma, and plasma is a part of extracellular fluid. This all we know. So with this much knowledge, let's approach some questions. Question reads: total water contained in body of adult male weighing 70 kg is 24 liters, 42 liters, 14 liters, and 28 liters. Answer is 42 liters. Why? 60% of total body weight is water. So answer will be 42 liters. There are two other numbers which are important: 14 liters of ECF, extracellular fluid, and 28 liters of intracellular fluid. I hope this concept is clear to you. Let's move to the next multiple choice question. Intracellular cation is, we have already seen there is ICF, there is ECF, and you have studied it throughout here. So major intracellular cation is it sodium, calcium, potassium, or magnesium? So major intracellular cation is potassium. Now there is also a small debate if major word is not used, that magnesium is also more in ICF as compared to ECF. So correct word will be major intracellular cation. Is it clear? Sodium is major extracellular cation. So more amount of sodium is present outside, more amount of potassium is present inside. At rest, the gradient, of potassium is more. So concentration gradient for potassium is more. That's why more amount of potassium always moves out at rest when cell is not stimulated as compared to sodium moving in. This movement in is called as influx, movement out is called as efflux, and that forms the basis of ionic basis of action potential. That will come subsequently. Let's move with the next multiple choice question. False about membrane potential is. Option A reads, resting membrane potential is more in smooth muscles as compared to cardiac muscles. Option B, when membrane potential becomes positive than resting membrane potential, it is called as depolarization. Sodium potassium ATPase pump plays vital role in maintenance of resting membrane potential. And fourth option is, when membrane potential becomes more negative, the cell is always inhibited. This is a long multiple choice question, usually a concept testing multiple choice question. What will be the answer? False statement is, when membrane potential becomes negative, the cell always is inhibited. Now, this portion you will not find in first or second chapter, but this concept you will follow whenever you start reading about special senses and CNS. So, rods and cones, when they undergo dark and light adaptation, they are stimulated even when they undergo hyperpolarization. Hyperpolarization is whenever membrane potential becomes more negative. Okay, resting membrane potential of smooth muscles is minus 56, which is higher as compared to cardiac muscle, which is minus 90 millivolts. Membrane potential becomes positive, that is movement away from polar state is called as depolarization. Regaining polar state is called as repolarization. So there are two phases of action potential: depolarization and repolarization. Sodium potassium ATPase pump, yes, it is a functions of sodium potassium. Potassium pump is, I tell you why it is necessary, because it is said, it's hypothesized that 50% of your energy requirement, 50% energy requirement is for maintenance of sodium potassium ATPase pump. Is it fine? So answer here is, when membrane potential becomes more negative, cell is always inhibited is a false statement. Let's go ahead. Now let's talk a bit about transport across cell membrane. Facilitated diffusion dash is non-saturable. Hello. I guess there was some problem. Now I'll share the screen again. And we were at facilitated diffusion. Yes, sir. Hello. आता झालंय सर, सुरू करतो मी.
Okay,
I'll
okay.
begin.
Yes, sir.
Huh. Facilitated diffusion. Next multiple choice. There was some technical issue, it seems. We'll begin with the next multiple choice question. Facilitated diffusion is dash. Is non-saturable process. Is faster than simple diffusion. Does not require special carriers. Does not experience competitive inhibition. You have to find out the correct statement amongst these four. And what is the correct statement? Facilitated diffusion is faster as compared to simple diffusion. Yes, it is a saturable process. It requires special carriers and it experiences competitive inhibition. So, facilitated diffusion is a faster process as compared to simple diffusion. Is this fine? Everybody is okay with this? Oh, now next thing. Now we'll be with nerve and muscle physiology, and that is another favorite area of most of the physiology teachers. And in this, structure and classification of neurons, especially Erlanger-Gasser classification of neurons, is important. Significance of myelination and conduction of impulse, how it happens, the saltatory conduction is an important topic. Nerve action potential, the ionic basis of that has always been asked as a short answer question, very rarely as long answer question. Synapse is the junction between two neurons, and this is a topic which is usually covered in central nervous system. So another is muscle component. So classification and structural characteristics of muscles. These things you have learned in 11th, 12th standard also. You know there is skeletal muscle, there is a smooth muscle, there is a cardiac muscle. Cardiac and skeletal muscle are striated muscles, whereas smooth muscles are non-striated. They're spindle shaped. uh, Cardiac and smooth muscles are involuntary, whereas skeletal muscles are voluntary. Cardiac muscles possess intercalated discs, which are nothing but gap junctions, and that is why the whole cardiac muscle functions as one single unit. And this property of cardiac muscle to function as one single unit is called as syncytium. It is called as syncytium. Then next topic is skeletal muscle contraction and excitation contraction coupling. Excitation contraction coupling has always been asked as short answer question. Every second or third examination, you will find this question. And what is the most important thing you have to remember in this? What is the excitation contraction coupler? So calcium is the coupler between excitation and contraction. So as a result of excitation, there is release of calcium from sarcoendoplasmic reticulum. This calcium goes and binds to C subunit of troponin, and then there is production of contraction. The whole thing you have to know. What is most important thing? Excitation contraction uncoupler or coupler, excitation contraction coupler is calcium. Then neuromuscular junction. Recently, this question has always featured in MUHS examination. You need to know how to draw good diagram and explain the functioning of neuromuscular junction. In neuromuscular junction, most important question that is asked is what is a neurotransmitter in neuromuscular junction? And disease of neuromuscular junction and applied aspect of neuromuscular junction is myasthenia gravis. Let's see this. This is a question based on Erlanger-Gasser classification. Muscle spindle organ receives motor supply through dash dash. Muscle spindle organ are present in intrafusal skeletal muscle fibers. These are specialized skeletal muscle fibers which are playing role in sensing and maintaining muscle tone. You might be learning about stretch reflex later on. So stretch reflex is mediated by a muscle spindle. So muscle spindle receives motor signal from which type of neuron? Which type of neuron as per Erlanger-Gasser classification? A alpha, A beta, A gamma, or A delta? Answer here is A gamma. So A gamma is motor to intrafusal skeletal muscle fibers. Intrafusal skeletal muscle fibers are nothing but muscle spindle organ. Okay. What is the function of A delta then? A delta carries fast pain. There are two components of pain. One is fast pain, the other one is slow pain. Slow pain is carried by C fibers. Is it clear? How this Erlanger Gasser classification is made? It is based on degree of myelination, diameter of neurons, and velocity of conduction. What we need to know, the myelination. This myelination aspect is function of Schwann cells in peripheral nervous system, whereas it is function of oligodendrocytes in central nervous system. Important multiple choice question. Schwann cells in peripheral nervous system, oligodendrocytes in central nervous system. This is Erlanger-Gasser classification: A alpha, A beta, A gamma, A delta. You might be seeing proprioception and somatic motor are functions of A alpha, A beta. Most of the neuronal receptors present in skin, like Pacinian corpuscle, Meissner's corpuscle, these are modified A beta fibers. These are all modified A beta fibers. A gamma, we have already seen the multiple choice question on that. A delta, as I told you, carries fast pain. C carries slow pain. Please remember this. Sometimes question is asked which has got least velocity of conduction. So if you have to know what has maximum velocity of conduction, so A alpha has got maximum velocity of conduction, and C type has got least velocity of conduction of the degree of 0.5. The degree of 0.5. Is this fine? Let's move ahead. There is another mode of classification called as numerical classification, and it is applicable only for sensory neurons. Numerical, Roman numerical numbers are used. 1, 2, 3, 4. So type first A, type second A, type third, like this. Clear? What is important? Golgi tendon organ. This word you will find over here. What is Golgi tendon organ? It is going to mediate the process of inverse stretch reflex. What is that? Inverse stretch reflex. This will be a part of central nervous system. But just remember that it is opposite to stretch reflex. So it is going to counter muscle spindle. Golgi tendon organ counters muscle spindle. Is it fine? Okay. And the fast pain component I told you, A delta carries fast pain. This is one favorite topic of some examiners. Which neurons are most susceptible? Which neurons are most susceptible for local anesthetic agent? Unmyelinated neurons, C type of neurons, are most susceptible for local anesthetic agents. Please remember this. Is it clear? Okay. Then let's consider the next one. Which of the following excitable tissue undergoes depolarization as a result of influx of calcium ions? So if you remember a bit of physiology, then usually it is entry or influx of sodium ions which is going to cause depolarization. In neurons, there is entry of sodium which causes depolarization. In cardiac ventricular muscle, there is entry of sodium which causes depolarization. Same applies to Purkinje fiber. What is different? It is smooth muscle which is different. In smooth muscle, spike potential or action potential is triggered by entry of calcium. So answer here is smooth muscle. Which is the other tissue where you will find similar thing? SA node is the tissue where you will find similar thing. There are two types of potential seen in smooth muscle. One is called a slow wave potential. The other one is called a spike potential. Slow wave potentials are not action potentials. They are local potentials, and they are because of sodium. They are because of influx of sodium and not because of calcium. Is this fine? This is brief review of action potential in neuron. This black one is action potential in neuron. This red one is going to depict changes in sodium movements, and this violet one is going to depict changes in potassium movements. You might be seeing that duration is very short, 2 to 2.5 milliseconds, and that is why the refractory period is also very small, very short. It is approximately 1 to 1.5 seconds. This is another basis for important multiple choice question, that neurons or skeletal muscle fibers, they can show tetanizability, but cardiac muscle does not show tetanizability. Why? Because it has got long refractory period. This is cardiac muscle which has got long refractory period. Just look at this, 200 milliseconds. There it was 2.5 milliseconds. This is 200 milliseconds is the total duration of action potential. And out of that, almost 100 to 150 milliseconds is refractory period. So it has got very long refractory period, and that is why it is having a property of non-tetanizability. This phase 2 is a peculiar feature of cardiac or Purkinje fiber action potential, and this is called as plateau phase, and it is because of simultaneous opening of calcium and potassium channels. This is another topic which is usually asked. Now this is a comparison between cardiac ventricular muscle and SA node. Just pay attention over here. Look at the resting membrane potential. It is minus 90 here, and it is almost minus 55 here. So SA node has got higher resting membrane potential, and it is very excitable tissue as compared to this one. Now this slanting portion is usually called as prepotential, or it is also called as pacemaker potential. This pacemaker potential is because of funny sodium channels. Pacemaker potential is because of funny sodium channels. The phase of depolarization, that is phase 0, is because of movement of calcium, and phase 3, that is phase of depolarization, is because of movement of potassium. This difference is very important in Viva, so you should prepare this very well. Prepotentials are because of funny sodium channels. Neurotransmitter at neuromuscular junction, as I have already mentioned, the answer here is acetylcholine. Answer here will be acetylcholine. And then you have to know about the structure of neuromuscular junction. What happens here? Just pay attention over here. Calcium is essential for exocytosis of vesicles. That's why whenever a nerve stimulus comes, this is A alpha neuron. This is motor neuron. This is A alpha motor neuron, and it causes entry of calcium in axonal terminal. This calcium causes exocytosis of vesicles containing acetylcholine. Acetylcholine is released. It goes and binds to nicotinic muscle end plate type of cholinergic receptor, NM type of receptors. Okay, and this is how the signal is transmitted. This is a classical example of neurohumoral transmission. Hmm? This is neuromuscular junction, an important question, important short answer question. A disease of neuromuscular junction is myasthenia gravis. It is autoimmune disease in which there is development of autoimmunity against NM type of receptors, and there is progressive muscular weakness. Skeletal muscle contraction, as we have already seen it briefly, that role of calcium goes and binds to troponin, C subunit of troponin. Here it goes and binds. There is conformational change, and there is exposure of myosin binding site. Myosin is in cocked up or charged state because of binding of ATP, and only in this state there is a power stroke. This is called as power stroke. Whenever you have to have detachment of myosin head, another ATP comes, binds here, and there is detachment here. So contraction as well as relaxation requires ATP. Both are active phenomena. Contraction of skeletal muscle as well as relaxation, both are active phenomena because they require ATP. And on this concept, requirement of ATP, this concept, contraction remainder, a concept is all often asked in Viva, or a concept of, yes, any one of you? Rigor mortis, yes, very important. Rigor mortis is very important. So study about that. Next system will be blood. I hope it is not too fast for you. Okay, am I rushing into the things? Just let me know. Okay, composition and function of blood. Plasma proteins are very important Short answer question. Erythropoiesis, another very, very important short answer question as well as long answer question. It can come as both. And blood is anyway a very important topic in practical physiology. So you have to know everything in and out about blood. The WBCs, types of various WBCs, and immunity and its classification. Hypersensitivity reactions, there are type 1, type 2, type 3, type 4. What you need to know that anaphylactic reaction is type 1. It is IgE mediated hypersensitivity reaction. Functions of platelets, we often tend to miss this question and can come as an odd short answer question. Functions of platelets, very rarely been asked, but it is very important. Hemostasis, another very, very important, mark it very, very important as long answer question or short answer question. Hemostasis includes both. Hemostasis includes both formation of temporary as well as permanent hemostatic plug. And before that, there is vasoconstriction. So, three phases: vasoconstriction, formation of temporary hemostatic plug, and formation of permanent hemostatic plug. Formation of permanent hemostatic plug means the whole cascade of coagulation you have to explain. Then there are blood groups, blood transfusion, very important topic, and erythroblastosis fetalis, a very frequently asked short note. Let's proceed with some questions. All plasma proteins can be synthesized by liver except, we all know most of the plasma proteins are synthesized by liver except, answer here is C, gamma globulin. What are gamma globulins? Gamma globulins are antibodies, Ig, IgG, IgM, IgA, IgE, and IgD. Functions of all of them you need to know. IgG is the smallest one and it can cross through placenta, and that is the basis for development of erythroblastosis fetalis. What is the first antibody formed as a response to a new antigen? It has to be IgM. IgM is always the first antibody formed as a response to a new antigen. Whenever there is second exposure or re-exposure to any antigen, you may not find that there is formation of IgM. There will be straight away formation of IgG because of presence of memory B cells. Memory B cells, plasma cells are modified B lymphocytes. So memory B cells will straight away start forming. They will straight away start forming IgG. Sometimes this concept is tested. Next question. A very, very favorite Viva question, very favorite multiple choice question. Hemoglobin appears in erythrocytes at what stage of erythropoiesis? So, answer here is intermediate normoblast stage. It appears. Okay, it appears. Please remember this. What is the significance of reticulocyte? Reticulocyte stage is a stage at which RBCs enter circulation. So normally you will find reticulocytes along with erythrocytes in circulation. How much is the contribution of reticulocytes? It is usually 1% or less than 1%. So out of total RBCs, approximately 1% will be reticulocytes. If you have more number of reticulocytes in circulation, that means there is increased erythropoiesis. And usually you will find increased erythropoiesis whenever there is increased destruction or there is low hemoglobin. So in case of anemia, you will get increased reticulocyte. Whenever you start the treatment for anemia, reticulocyte count will decrease. So it has got prognostic value. Sometimes this question is asked that it has got prognostic or diagnostic value. So it has got diagnostic value but more of prognostic value. Okay. Next question. Which of the following condition may not show presence of hypochromic microcytic anemia? What will determine hypochromicity? MCH will determine hypochromicity, mean corpuscular hemoglobin, or simply the appearance of RBCs on peripheral blood smear. They are pale looking, then hypochromic. Microcytic, small size. Okay, small size of RBCs. What determines the size? The mean corpuscular volume. So mean corpuscular volume decreases, it is called as microcytosis. Increases, it is known as macrocytosis. Macrocytosis is usually seen in cases of megaloblastic anemia. It is seen in cases of megaloblastic anemia. And what are various causes of megaloblastic anemia? B12 deficiency, folic acid deficiency, and your favorite intrinsic factor deficiency that causes megaloblastic anemia. So it causes macrocytosis and not microcytosis. What is this intrinsic factor? Intrinsic factor of Castle is secreted by parietal cells of gastric mucosa, and they are important for absorption of B12 in ileum. If there is an autoimmune disease which will cause destruction of parietal cells of gastric mucosa, there will be reduction in intrinsic factor, and this is called as pernicious anemia. Please remember, pernicious anemia is autoimmune disease. So whenever in Viva causes of anemia are asked, and if you utter pernicious anemia as the first answer, usually examiners will get very angry, because in India, the commonest type of anemia is not pernicious anemia. It has to be nutritional deficiency anemia, especially iron deficiency anemia. Please make a note of it. Whenever you are answering in Viva, do not say pernicious anemia is the commonest cause of anemia. No. First cause has to be iron deficiency anemia in India. Next question. Yes. Which of the following, this is a question on coagulation, which of the following coagulation factors are dependent on vitamin K for their activation? So which of the following is vitamin K dependent coagulation factor? So there is simple mnemonic: 2, 7, 9, and 10. These are four factors which are dependent on vitamin K. Amongst these, which one is factor 2? Anyone? Factor 2, factor 2? Yes, factor 2. This is factor 1, this is factor 8, this is factor 13. So answer here will be prothrombin. So please remember about vitamin K dependent coagulation factors. And you should know about intrinsic and extrinsic pathway of coagulation. What is the significance of prothrombin time? What is the significance of activated partial thromboplastin time, APTT? So please read about these things. Hmm? Next. Yes, this is a very long multiple choice question, and it will take a while to read this. I'll read it out for you. Which of the following is true about blood transfusion? A. Transfusion of large volume of stored blood causes right shift of oxygen hemoglobin dissociation curve. This is a topic from respiratory system, but very closely related to blood. So I have coupled this. So transfusion of large volume of stored blood causes right shift of O2 hemoglobin dissociation curve. During major cross matching performed before blood transfusion, donors' RBCs are matched against recipient's plasma. Blood group ORH positive is called as universal donor. Transfusion of 1 unit of blood increases hemoglobin by 2 gram percentage or 2 gram deciliters. When you are reading such a long multiple choice question, you usually tend to get very irritated, very irked, and you start thinking something like this. These sacred words come out. What kind of multiple choice question has been framed? Don't get panic. Usually, such questions are very, very friendly questions. I can assure you that. Longer the question, more friendly it is, because everything is written in that. Hmm? So, what is the answer in this? Correct answer is major cross matching. In that, donors' RBCs are matched against recipient's plasma. What is minor? Donors' plasma and recipient's RBCs. Another very important question. Transfusion of large volume of stored blood causes left shift. Why will it cause left shift? Because stored blood has got lower concentration of 2,3 bpg. So you need to know about various factors affecting oxygen hemoglobin dissociation curve, causing right shift, causing left shift. Another very favorite topic, very frequently asked topic, oxygen hemoglobin dissociation curve can come as short move. So please prepare that. Blood group ORH positive is not universal donor. Blood group ORH negative is universal donor. And transfusion of 1 unit of blood increases hemoglobin by 0.8 to 1 gram deciliter. Hmm? So it increases by 0.8 to 1 gram percentage. Single unit of blood, single bottle of blood, single bag of blood, if it is given to a person, it will increase hemoglobin by 0.8 to 1 gram percentage. Is this clear? Next system will be respiratory system. Now we are left with respiratory system, renal, and GIT, and we have approximately 20 minutes time. So let's rush through. Let's quickly go through these systems. Divisions of respiratory tract and concept of dead space, very important. Muscles of respiration, we know chief muscle of inspiration is diaphragm, and it is assisted by external intercostal. Muscles of expiration are abdominal muscles and internal intercostal. Mechanics of respiration, the two important movements, bucket